स्टूडेंट्स, वेलकम टू बायोमिक्स। आज के इस वीडियो में हम डिस्कस करने वाले हैं प्रोटीन टारगेटिंग इनटू माइटोकॉन्ड्रियल मैट्रिक्स। ये जो सेल ऑर्गेनल जो प्रेजेंट है माइटोकॉन्ड्रिया, इसका खुद का प्रोटीन, इट इज हैविंग द एबिलिटी दैट इट कैन सिंथेसिस इट्स ओन प्रोटीन। ये खुद का प्रोटीन का सिंथेसिस कर सकता है। दिस माइटोकॉन्ड्रिया इट इज हैविंग 13 डिफरेंट टाइप ऑफ प्रोटीन्स। स्टिल देन ये डिपेंड करेगा हमारा न्यूक्लियस पे for some of the proteins. Why? Because this mitochondria, ये जो cell organelle mitochondria जो present है, it is doing different type of work. So what are the works being done by this mitochondria? यहाँ पे हो रहा है oxidative phosphorylation is taking place in the mitochondria, as well as ATP synthesis is taking place in the mitochondria, and electron transfer system is also taking place in the mitochondria. So it requires different type of proteins and it depends upon the nucleus for some of the proteins. So ये जो protein जो present है, it is having some of the signal sequence with it, so that it is having the ability that it can transfer this protein into this organelles. And this mitochondria, ये जो mitochondria जो present है, इसका different layers present होता है. Like it is a double membrane, it is having a double membrane with it. इसका जो बाहर का layer जो present होता है, mitochondria का जो बाहर का layer जो present होता है, it is known as outer membrane. And inside there is another one membrane which is known as इनर मेम्ब्रेन. ये जो प्रेजेंट है, ये इसको बोलते हैं हम इनर मेम्ब्रेन. एंड ये दोनों के बीच में, आउटर मेम्ब्रेन और इनर मेम्ब्रेन के बीच में जो स्पेस जो प्रेजेंट है, दैट इज नोन एज इंटर मेम्ब्रेन स्पेस. इट इज नोन एज इंटर मेम्ब्रेन स्पेस. Okay, and inside the inner membrane, the space which is present, it is known as matrix. Okay, so for transferring a protein into different type of layers, we can tell that this protein can be transferred into intermembrane, it can be transferred into outer membrane, it can be transferred into inner membrane as well as matrix. It depends upon that the protein, where the protein is being required. सो आज के इस वीडियो में हम डिस्कस करेंगे कैसे एक न्यूक्लियर प्रोटीन हमारा माइटोकॉन्ड्रियल मैट्रिक्स को टारगेट करके it will go inside the matrix. Okay, so when we discuss about the protein, जब कभी भी हम एक प्रोटीन के बारे में डिस्कस कर रहे हैं, उसके पास कुछ सिग्नल सीक्वेंस प्रेजेंट होगा. So this signal sequence which is present for the mitochondrial matrix, it is in the N-terminal end. And in this N-terminal end, it is having 15 to 55 amino acids which are present. They are also known as pro-sequence. Okay, and it is also known as MTS. What do you mean by MTS? MTS का मतलब क्या है? It is known as mitochondrial targeting sequence. It is known as mitochondrial targeting sequence. Okay, so ये जो यहाँ पे जो अमीनो एसिड्स जो प्रेजेंट होते हैं, they are mostly positive amino acids which are present for targeting the mitochondrial matrix. So ये जो mitochondrial जो matrix में जो targeting होगा, what is the type of targeting? Already we have discussed about the post-translation targeting as well as co-translation targeting. So ये जो mitochondria में जो protein transfer होगा, it will be transferred post-transcriptionally. Post-transcriptionally का मतलब क्या होता है? It means that after the complete synthesis of the protein, it will be targeted to the organelle, that is the mitochondria. Okay, so let us discuss how it is being transferred. So यहाँ पे हम देख सकते हैं एक प्रोटीन का पूरा सिंथेसिस हो चुका है. Okay, when this protein is being synthesized, to prevent it from misfolding, अगर एक प्रोटीन का सिंथेसिस पूरा हो चुका है, then it will help in the folding of the protein. So for preventing the protein from misfolding, there are certain chaperones which are present. और ये जो chaperone जो present है, it will bind in the protein. Okay, and it will help in preventing the protein from misfolding. और ये जो प्रोसेस है, इट इज ए एटीपी डिपेंडेंट प्रोसेस. यहाँ पे एनर्जी का रिक्वायरमेंट हो रहा है. ओके, सो यहाँ पे हम देख सकते हैं एन टर्मिनल एंड पे प्रेजेंट है हमारा माइटोकॉन्ड्रियल टारगेटिंग सीक्वेंस, एंड दिस माइटोकॉन्ड्रियल टारगेटिंग सीक्वेंस विल बी रिसीव्ड बाय वन रिसेप्टर विच इज नोन एज इम्पोर्ट रिसेप्टर. ओके, एंड दिस विल बी प्रेजेंट इन द आउटर मेम्ब्रेन, और इसे हम क्या बोल सकते हैं? इट इज नोन एज TOM 20 or TOM 22. Okay, after receiving this protein sequence by the receptor, it will transfer it into the translocon. Okay, so this translocon which is present, it is known as TOM40. TOM40. Okay, it is also present in the outer membrane. Why it is known as TOM? It is known as TOM because it is present in the outer membrane. Translocon, it is known as translocon of outer membrane. Okay. After this set of protein is being transferred into this translocon, it will be targeted to the translocon which is present in the inner membrane. Okay, and this is also having one receptor which is known as TIM14. This TIM14 which is present, it is a receptor. Okay, and TIM23 and TIM17 which is present, they are the translocon. They are the translocon which is present in the inner membrane. Okay, inner mitochondrial membrane. So what it does, it helps in transferring this protein from the outer membrane inside the mitochondrial matrix. Okay, so यहाँ पे भी क्या प्रेजेंट है, यहाँ पे भी जो प्रेजेंट है इट इज, यहाँ पे जो प्रेजेंट है यहाँ पे भी प्रेजेंट होता है कुछ चैपेरॉन्स विच इज नोन एज मैट्रिक्स चैपेरॉन. और ये मैट्रिक्स चैपेरॉन्स का भी नेम क्या है, इट इज द HSP70. ये जो HSP70 जो प्रेजेंट है, व्हाट इट डज, इट हेल्प इन प्रिवेंटिंग फ्रॉम मिसफोल्डिंग एज वेल एज इट विल हेल्प इन पुल्लिंग द प्रोटीन. Okay, it will help in pulling the protein from the inner membrane to the matrix. Okay, after this protein has reached into the matrix, it will be recognized by one protease which is known as mitochondrial processing protease, or it is also known as MPP. So what is the work of this MPP? It will cleave the sequence, it will cleave the targeting sequence. Okay, after the cleavage of the targeting sequence, this sequence will be degraded. Okay, and now with the help of this HSP70 which was a chaperone, it will be completely pulled, and this protein will be released into the mitochondrial matrix and it will be folded. When the protein is in the folded form, then it can work whatever the protein is targeting, it can work. Another one thing which I have forgot to tell you is that here the HSP proteins which are present before it is being transferred into the outer membrane, it needs to be removed because there is no need of this chaperones in this region. Okay, because as it is being transferred, as it is targeted into the TOM complex, it does not require any HSP proteins. So this removal of this HSP protein is also by this, it is a energy dependent process. Okay, let us see with this other diagram how it is being done. Like we can see here, there is a signal sequence in the protein and it has been recognized by a TOM complex which is present in the outer membrane of mitochondria. After that, it is being transferred. Here you can see here the recognition is taking place, and after the recognition, it has been targeted to the inner membrane by the help of one protein which is known as TIM23. It is a translocon. This TIM23 which is present, it is a translocon which is present in the inner membrane. Okay, ये जो present है TIM23 जो present है वो inner membrane में present है, और after it is being synthesized, after it is being transferred, ये जो present है it will be released in the mitochondrial matrix. Okay, then it will be cleaved. ये जो present है इसका signal sequence present है यहाँ पे, तो ये cleave हो जाएगा हमारा mitochondrial matrix पे और एक mature mitochondrial protein का formation होगा यहाँ पे mitochondrial space पे. Okay, so another thing which you have to understand is ये जो present है यहाँ पे जो sequence जो present था, we have already discussed that in the N terminal end there are positive amino acid sequence which are present. So यहाँ पे जो present है हमारा mitochondrial matrix का space, mitochondrial matrix का जो space जो present है, it should be negative. क्यों ये negative होना चाहिए? Because as this N terminal is having the positive amino acid sequence, so for attracting this positive amino acid sequence, the mitochondrial matrix space should have negative sequence. ओके सो ये जो माइटोकॉन्ड्रियल मैट्रिक्स स्पेस पे जो नेगेटिव सीक्वेंस जो प्रेजेंट है, व्हाई इट इज सो, इट इज ड्यू टू इलेक्ट्रोकेमिकल, ये जो नेगेटिव सीक्वेंस जो प्रेजेंट है माइटोकॉन्ड्रियल मैट्रिक्स पे, इट इज ड्यू टू इलेक्ट्रोकेमिकल, इट इज ड्यू टू इलेक्ट्रोकेमिकल ग्रेडियंट, ठीक है। सो जो है NADH FADH2 जो प्रेजेंट होता है हमारा मैट्रिक्स पे, व्हेन इट इज ट्रांसपोर्टेड एट आउटसाइड, ड्यूरिंग दिस टाइम देयर इज रिलीज ऑफ H+, due to which the outer membrane is positively charged and the matrix which is present, it is negatively charged, which also help in targeting the protein inside the matrix. Okay, here you can see that here in the protein, this is the protein, and in the Tom complex it is recognized, this signal sequence has been recognized with the Tom complex, and here there are some of the chaperones which are present, which will help in preventing the protein from misfolding. और यहाँ पे क्या हुआ, यहाँ पे जैसे कि यहाँ पे आउटर मेम्ब्रेन पे ये सिग्नल सीक्वेंस आकर बाइंड किया, देन इट इज बीइंग ट्रांसपोर्टेड इनटू द इनर मेम्ब्रेन विच इज नोन एज स्टीम 23. ओके, और यहाँ पे वी कैन सी इन दिस रीजन दैट इज पॉजिटिव सीक्वेंस एंड इन दिस रीजन दैट इज नेगेटिव सीक्वेंस, एंड here we can see that another HSP70 is present, and this is utilizing ATP to bring the protein inside. Okay, and this process which is present, it is an energy dependent process. Okay, I hope you have understood how this protein is being targeted into the mitochondrial matrix. In the next video we will discuss how the protein is transported into the outer membrane of mitochondria. Thank you everyone for watching this video till the end. If you like this video, please do not forget to subscribe. Thank you.